We're going to start off with a practice genetics problem just to see kind of where you're at. I'm assuming, Sarah, this is Sarah Eider. You've been doing this for weeks now. Do you really need genetics help? That's what we're reviewing. If there's something specific you want to go over this morning, we should probably do that first before going into basic genetics. But if you're just...
No, I don't. Yeah, I think I'm good. I was just like gonna join just cause.
Okay, I'm like, dude, you've been doing genetics for a couple weeks now. I feel like you're always present and I always see you, which is awesome. But if you don't need the help, don't feel like you need to, necessarily stay on the whole time. We're gonna do a basic problem because we haven't gotten into complex genetics for biology just yet. So we're gonna do some sort of a basic problem, but this one's gonna be, it's from the workbook. I don't know if I actually assigned this page in the workbook or not for you guys, but this is from the workbook and it's a basic problem, like a 4 squared Punnett square, but they're backwards problems. So they're a little bit of like a sleuth work, if you will, where they ask us to like figure out who parents are based on who their children are. So we're gonna go ahead and use some of these as our warm-up and then go on from there. I hate not being able to like talk to you guys and ask you questions. That's what I'm used to doing. So at this point, you've all tried the basic genetic stuff. I'm going to assume that you know how to do like a pretty simple basic 4 squared Punnett square. And so what I thought we would do is just reiterate some of the vocabulary that comes up when you're doing genetics problems, and then kind of make use of those as much as we can while we review today. So I'm going to mirror my iPad. Here we go. And if you're curious what page this came from, this is page 86 in the workbook for bio. So I'm going to go ahead and write on my whiteboard with my iPad here. So again, these are backwards. So they basically start you off with the offspring's outcome, and they're asking us to turn around and work our way backwards to figure out what the parents' genotypes would have been. So that's one of those vocabulary words, the genotype, and then they want us to figure out based off the genotype ratio. Okay, so a genotype versus a phenotype. So you gotta remember that when we make keys for genetics problems, one side of the key has those letters that we use for symbolism purposes, and the other side of our key gives us what that will physically give us in terms of a trait. So let's say we were looking at like the type of coat of a Labrador Retriever or something. Maybe these are rough coat Labrador Retrievers and these are smooth. The left, go ahead.
You didn't share your screen.
Oh, I didn't?
No.
Shoot. Well, that's not helpful. Let's try that again. Okay. So this is the page, page 86, and the can you do genetics backwards piece. And then I highlighted genotype right there because that's what we are being asked to include, is the genotype of the parents. And so what I was saying earlier is when you make a key, on one side you've got the genotypes, and those are these right here. And please understand, you obviously do not have letters inside of your body. These are just symbolic. They are symbolic of what types of A's, T's, G's, and C's an individual has on their chromosomes. 1 chromosome from mom, 1 chromosome from dad. Hold on one second, it says that there's somebody in the waiting room. Maybe not. Good morning, Jack. Sorry we missed you there. I didn't realize you were hanging out in the waiting room. Okay, so the next thing we're gonna do here is go ahead and identify these. And these are the actual protein, the actual proteins that are made as a result of the genetics. So since we've already done protein synthesis, if you were with us during enrichment, you've seen the process of protein synthesis, or that wasn't enrichment, that was actually last week. So now you've got an idea of what a phenotype is. Phenotype would be the physical features that result. So long story short, we're going to come back up here and do this backwards problem. So they want us to figure out what the parents would have to be if we have a 1-to-1 ratio of offspring. So kind of breaking that down, I'm trying to make this bigger for you guys. 1 second. Kind of break this down. The first number is always going to be whatever you happen to have the homozygous dominant outcome. Okay, the second one is always going to be your heterozygous, and your third one is always going to be your homozygous recessive. So once you have done enough of these, it becomes pretty easy to figure out what would produce this. In the meantime, we can kind of just fiddle. So I'll start with a parent who would have a dominant allele in that position, a dominant allele in this position, and then if I don't know the second letter, I'm just going to kind of leave it like that. We can fill in what we can, and let's see what that gives us. When you have an outcome like this, you got to think about, okay, what would you have? You would have one individual that's homozygous dominant there, and then if these were little r's, then these two would be heterozygous, and that's exactly what they're looking for in this ratio. And then the children in this last box would be homozygous recessive. So right off the bat, 2 heterozygous parents, which is what I've laid out here, they do give me the ratio that was requested. Now, if we bounce to the next one, they want to see, well, what kind of outcome would give us 4 of the homozygous dominant individuals. Well, the only way you're going to get nothing but homozygous dominant individuals is if all the parents also had homozygous alleles, or sorry, had dominant alleles. So parent number 1 would have to be completely dominant, and parent number 2 would have to be completely dominant. And therefore, that's all you get, right? So it's one of those things where after a while, you've done enough of these that you're like, yeah, I can do this in my sleep pretty much. And you already know ahead of time exactly what the parents' genotypes are going to be. But you may not be there yet, so that's fine. Now in this one, they want us to combine to produce 4 offspring that are all heterozygous. So the only way you'd have that is if one of the parents is contributing nothing but recessive alleles, and the other parent is contributing nothing but dominant alleles. So parent number 2 would have to be that combination. And then of course, if you fill that in, oops, you'll see, shoot, early in the morning, I make a lot of mistakes, sorry. You would have nothing but heterozygous children. Okay, and then the bottom, the fourth one is kind of probably the trickiest. 2 and 2, and then no individuals that are homozygous recessive. So we have to have an outcome that produces homozygous dominant individuals. I'm gonna guess, and I think this is gonna work. Perfect. Okay, and it's kind of like a guess and check type of thing, I suppose, when it gets down to this. And not everybody has the stamina to want to stick this out because it becomes kind of annoying after a while. Genetics is one of those things you either like it or you hate it. Now they want us to identify genotypes of parents, and we're looking at phenotype ratio that they've shared with us instead of genotype this time. So now we have to change our thinking. So we're looking at physically what would these individuals look like. So let's think about the golden retriever or Labrador retriever example. Let's consider rough versus smooth fur. So we come over here, and in the first one, it's all smooth fur. So the only way you would get nothing but smooth furred offspring is if the parents themselves were also smooth. That's what we would call a purebred, so a purebred individual. Purebred is just a fancy way of saying homozygous. And so that's another vocabulary I've been using is homozygous versus heterozygous. And hopefully you're kind of getting used to those two terms. If you're not, just to kind of break those terminologies down, you probably are familiar with other words that have similar beginnings or the same beginning. Homo refers to same and hetero refers to different or opposite. And then the zygous ending on these vocabulary words for genetics refers to a zygote. A zygote is the first type of cell that forms after the sperm and the egg unite. So zygous is in reference to what the offspring would be, basically. So they are either going to have two of the same kind of allele or two different kinds of alleles. My iPad just kind of froze up. All right, let's see. Hold on, I'm going to try and open that right back up again. Shoot. Hi, Gabe.
Hi.
All right. So Gabe, we're just reviewing one of the pages in the workbook as our opening today, and I'm kind of just throwing some stuff at the group. We're on page 86. I don't know if you did that page already, but that's where we are. Okay, and so the next one, let's do the one that's F, 2 and 2. 2 and 2 for the phenotype. So we want 2 rough-coated Labrador retrievers and 2 smooth. So we would have to have 2 individuals that have to have the dominant allele. And I don't know where they're going to show up in the Punnett square, so I'm just going to put 2 in there. And then we have to have 2 individuals that have nothing but recessive alleles. So we got to consider where each of those would have come from. Okay, and then the capital R had to have come from one of the parents. And then in this case, that parent would have to have a recessive allele as its second allele. And the only way that would work out then is if the 2 rough-coated dogs are heterozygous for the trait. There isn't a way that we could have had a dominant allele in this position and still had the outcome of a 2 to 2, because as soon as you add that dominant allele there, now that bottom square is no longer a smooth coated retriever, it's a rough coated retriever. So that wouldn't work for this scenario. Let me backtrack and go back to what is correct. And that is that the top parent or one of the 2 parents has to be homozygous recessive. So the outcome would be that is one parent and that's the other parent. 3 to 1 is a pretty common genotype to see. And so what I know we're going to see is a homozygous dominant individual there, a recessive individual there, and a homozygous recessive there. So here are your 3 rough-coated dogs, and here's your smooth dog right there. So your outcome is coming from parents that had the dominant allele there and the recessive allele in their second position. So each parent was heterozygous, I'm squished. And then the last one, H, a 4 to 0. So they're asking us how we would get nothing but rough-coated dogs. So you could have capital R's in all positions. And then regardless of what the second letter is, they're all going to be rough. Well, actually, that's a lie. No, that's correct. So the first parent has to have at least an allele that's dominant. Whatever their second letter is irrelevant. It could be a small, it could be a dominant allele. Either one is going to produce the same outcome here. And the same for the second parent. We don't even know what the second parent's genotype is. Could be dominant, could be recessive. We have no clue because it doesn't matter. That one parent has passed along its genes. And so physically, these are all going to be rough-coated retrievers. So I find this very helpful to do when you're unsure, is to do the little blank line. And that communicates not only to yourself, like I still have some unknown information, but it also communicates to someone grading your work or looking at your work that like, okay, you get that there's uncertainty there and that later maybe you could fill it in or maybe not. So in this case, we've got lots of options that could have resulted, okay? So what questions do you guys have, if any, about some of the things that I've been going over? Because I don't want to just talk at you. I'd love you guys to like ask for clarity and interrupt me. We're all good with what we just did? Okay, I'm good with that then. So the other type of problem we threw at you last week, sorry, I'm fiddling. The other kind of problem we threw at you guys last week was a dihybrid genetics problem. And those are uber fun. There's a lot to those. And so what we could do is take a look at some sample problems that I haven't assigned from the workbook. Maybe you've already tried them anyway. And then just kind of see what that process would look like and reiterate that dihybrid process. So a dihybrid problem is different from what we just did. What we were just doing are what are considered monohybrid problems. Monohybrid problems are when you're dealing with a single type of trait at a time. So monohybrid, mono means one in Latin or Greek. And so a hybrid refers to like the fertilization or the mixing or mating of two individuals. So we did those, and that produces the simple 4-box Punnett square. Dihybrid problems look at two traits at a time. So it's basically just a more advanced way of looking at what are the likelihood of inheriting this trait and this trait at the same time. And you got to consider what happens during meiosis in order to fully understand that. So if you guys did the enrichment activities with us a few weeks back, we looked at meiosis as a process. And one of the things that we learned is that chromosomes are inherited in pairs, one from the father of an individual and one from the mother of an individual. And I just drew those symbols opposite. And then we have 23 pairs of those inside of human cells. So what we're really meaning when we do dihybrid problems is what's the likelihood of a trait on this chromosome being inherited along with a specific type of trait on a different chromosome somewhere else in that individual's collection of chromosomes. So we're just looking at the odds of these two chromosomes basically being inherited together, more or less. So like if this one has a blue-eye gene on it and this one has a curly hair gene on it, how likely are we to pass along this particular chromosome and this particular chromosome into a single sex cell, and then pass that on with our partner to our offspring. So it's a little complex to explain, but basically that's what we're doing. So we're gonna go ahead and line up a problem. Might as well look at one that's in the workbook. If you have your workbook in front of you, I kind of picked and chose when I was making all the instructional videos and things for you guys. Let's do some stuff from page 101. Got a variety of different problems on it. So I'm going to go ahead and just select one. How about number 3 on page, no, number 3 is kind of done for us. Let's do number 4 on page 101. If you haven't done that yet, and you have your workbook, this is a good place to go. And then I also have a second one that we can use that I just photographed, number 5. So we'll see how it goes. Crop this. All right, and then let's make this bigger. I can see what's happening. Okay, that's good enough. All right, so when we are doing a dihybrid problem, just like regular genetics problems, you always want to start by making yourself a key. So I always read the problem first and figure out, well, what are the two traits that we're trying to figure out here? So looking at number 4, we're looking at werewolves. We're looking at fangs and whether they're sharp or round. And then the second sentence reads that we're looking at hair. Long hair is dominant, short hair is recessive. Okay, so they ask us to cross one type of parent with another kind of parent, and then they've set up a little bit of it for us, and they're asking us some questions about the offspring down low. So we're going to go ahead and make the key here off to the side. I'm going to kind of squeeze it in. Can you guys see that key? If you can't, let me know. On my screen, it's kind of squished. So we've got sharp fangs and round fangs, and the sharp are dominant, and they're asking us to use the letter F. So I'm going to go ahead and squeeze the capitals and the lowercase is in here. F is a funny letter because if you get sloppy or if you kind of don't have the best neat handwriting, a lowercase can start to look like a capital. So be careful and maybe put a little line over your F to differentiate what's capital and what's lowercase. And by the way, a lot of my biomed kids have been doing genetics problems too, and they've been typing out all of the Punnett squares and everything else. Oh my gosh, what a pain in the butt. Don't do that. If you can avoid typing them and just like snapping a photo of your work on paper, that's so much easier, I would think, than trying to type out and make boxes. And it just seems complicated. It's your choice, but this seems easier to me to handwrite it. Okay, so sharp is dominant. So that's going to be our first setup there, our second setup there, and then our third phenotype will be the round fangs. And then the second trait in question is hair, and so long hair is dominant, and they want us to use H. All right, and then short is the recessive. All right, so once we have the key set up, now we're going to figure out who we're crossing. They've asked us to cross a homozygous sharp fang with a heterozygous long hair. All right, so we're going to basically have one parent that is homozygous for sharp, so there'll be two capital Fs. You can always put it here too. They didn't give us a lot of room. And that same parent is heterozygous for long hair. So we'll put a capital and a lowercase side by side for that parent's genotype. So that's parent number one. And we're crossing that parent with a round fang short hair. So round fang short hair is going to be nothing but recessive alleles all the way across. Okay, so there's our two parents. We'll write them here as well so they're not so squishy. All right, when you're doing a dihybrid problem, the next thing to do is to figure out how you set up the Punnett square. So in this case, what we're really doing is figuring out the result of egg or sperm production during meiosis. So we're going to go ahead and use that FOIL method that I introduced, and you may know from math. In the FOIL method, which stands for first, outer, inner, and last, we're going to be combining different letters within each parent's genotype. So for example, if I look at parent number one here, we're going to take the first F and the first letter of the second gene, which would be the first H, and we're going to combine them and just put a little circle around them, demonstrating that we just made, let's call that the female werewolf, we just made an egg. And inside the egg, chromosomes that have the dominant F and the dominant H would have been pulled together during meiosis. In the outer, moving on, the outer F and the outer H are going to combine next, and we'll put those two together in a circle. Then we move to the inner I, and we're going to do our inner F and our inner H, which happens to be the same combination we had earlier. And then our last L and our last F and our last H are going to combine, and we can put a circle around those two. So if you don't like FOIL, you can do this on your own, fine. You don't need to use FOIL to figure out all the different combinations that could result. In the case of the second parent, there's not really a whole lot to figure out because they have nothing but a recessive alleles. So no matter what type of sex cell they make, they will always be combining a little f and a little h. So they're going to have 4 of those, okay, around the Punnett square. So let's go ahead and plug those into the actual Punnett square that's here on the paper. And so I'm going to put parent number 1 across the top. So capital F, capital H, and capital F, little h. And then the second and or the third and the fourth column repeat. Okay, and then the second parent will go down the side here. And because there is so much repetition, that means we're going to have a bunch of repetition within the Punnett square itself. So you can do some shortcuts when you fill in the Punnett square. Some of the more common places kids make errors is when they are filling in big Punnett squares. So you got to be kind of thorough about it. So if you do a shortcut, that's actually fine, but just make sure that you're correct to start with. So in this particular one, make sure you keep your F's together, keep your H's together. It just helps later when you're trying to figure out what this individual is going to look like, because you can refer back to your key and it's already written that way. So all 4 of that column is going to be the same. And then for the next one, this is a lowercase h or f, and I want to make that very clear. And then we have 2 little h's side by side, and all of those would look the same. And then the next 2 columns are going to repeat the first 2 columns. So we'll have that capital F, little f combination, and then capital H, little h, and then capital F, little f again, and this time little h, little h. All right, so out of the 16 boxes, 8 of them have this combo, and 8 of them have the other combo. So let's look at what those werewolves will look like. In the first column, these individuals are going to be sharp fang, and they're all going to have long hair. So we'll call that a sharp and long. And there's 8 of those within our Punnett square. I'm starting my ratio. I have 8 sharp and long. And then since it's a ratio, we have to put colons. The next one is also sharp, but instead they are short hair. So we have 8 of those as well. Okay, and for the most part, you're done. Technically, when you're doing ratios, you also have to include what is not there. If a team is just rocking their season, you don't just say that they have 8 wins. You always say they have 8 wins and 0 losses. So in this case, we should also put zeros in front of the combinations that aren't present here. So they don't have any offspring that are round fang and long hair, and they don't have any offspring that are round fang and short hair. So technically we should add that, and that makes a complete ratio. Round, long, and round, short. Okay, so that's a phenotype ratio. Ideally, we would have written it in one long strand, but clearly there's not enough room for that. The next question is just simply asking us about a specific type. So round fangs and long hair, the probability of that happening is 0. They will never have that combination. And then sharp fang and short hair, it was 8 of the 16 boxes, so that's half of the boxes, right? So you could say 50% would be the probable odds. You could say half, whatever you're comfortable, articulating. 8 out of 16, whatever, it's all the same thing. And that's basically it for the first problem. They're quite a bit involved. So if you want to do the second one, we're looking at beauty queens, clearly all made up problems. Long legs are dominant to short legs, and high cheekbones are dominant to average cheekbones. A man that has short legs and average cheekbones marries a woman who's heterozygous for both traits. So again, we'll set up a key. This is going to be for number 5. For our key, we're going to use L's for legs. And these are long legs versus short legs. And then the cheekbone business, they want us to use B. Average cheekbones are the recessive trait, and high cheekbones are the dominant trait. So our parents that we're going to be crossing will be a man that has short legs and average cheekbones marries a woman who's heterozygous for both traits. So capital L, capital B, little b. So as far as his setup goes, when we FOIL, we're going to get a really boring result, just like we had in the last problem. And so the husband will be kept little l little b all the way down here on the side. And it doesn't matter if the male or female go across the top or on the side. You just put one parent in one of those two places, the other parent goes in the other. And then for foiling the wife, we're going to do the combo first, outer, inner, and last. So that would be her setup. So then we come up to the Punnett square, fill all this in. And then work your way down. We got a little variety there. And then since the man is the same, all 4 boxes going down will be the same there. So we can do a little shortcut. And then they're asking us to plug in our ratio, and they gave us very little room to do that. So looks like for phenotypes, they will have the long-legged, high cheekbone variety, and that will be the first 4. So I'm just going to mark them off so I know I've counted them. Okay, so that's the 4 long and high, colon. The next 4 are going to give us long legs and average cheekbones. We're going to have 4 of each combo possible. Then the next 4 are going to be the short leg, high cheekbone. I hate when they give you no room. Short and high, and I'm going to go ahead and just continue the ratio up above. The last combination is 4 of them that are short legs and average cheekbone. Just double check you didn't do anything wrong there along the way. We've got all of them counted. Great. So then part B is asking us, based on known beauty queen characteristics, what's the probability of this couple having a future beauty queen? So we got to think about, they said that they didn't really tell you what a beauty queen is defined as, but knowing kind of life, right? The beauty queen is defined as someone who has long legs and high cheekbones. So we'll look to see, well, who has that? Again, that's a 4 out of 16 in the Punnett square. So that was a 25% average, 1/4. And the pregnancy that would come next would be no different, right? The Punnett square applies to all pregnancies. So just because they've already had a child, perhaps that already had those 2 characteristics, doesn't mean that they can't have another. So we would still look at the Punnett square for the future probable odds. And they would have a 4 out of 16 odds of having another, which equates to a 4th or 25% chance of having a child that had long legs and high cheekbones. And that's the end of that problem. All right. If you guys have any questions, go ahead and ask if you're comfortable. Talking to myself. Okay, so there's plenty of other problems that are in the workbook pages that I'm sure you could do. I made lots of answer keys, and I guess you checked a lot of them last week. If you didn't, please do that before you go and submit work. Like if I give you answer keys, check your stuff. I know that ship has sailed at this point. What's happening this week is getting into complex genetics. So you're going to start to change the rules. So rather than having some of these rules you've just seen here come into play, we're going to see different scenarios. And I'll give you a quick preview of what that's going to look like. Some of the different rules that might change would include things like no longer will a scenario such as this middle one produce, going back to the beginning of our talk today, if this was a rough-coated Labrador Retriever and this was smooth, right? what we were anticipating is that this allele dominates over this allele. Well, we're going to see that sometimes there is no dominance. So we might have a situation instead where instead of dominating over the other allele, they both make protein, and that the final protein ends up looking somewhere in between the two extremes. So instead of it being rough or instead of it being smooth, we have a dog who has fur that's somewhere in between the two. I don't know what that would be called. Roughish, smoothish. So it's not so rough, basically. Very scientific here, roughish, smoothish. That's hard to say. So when you see this kind of in-between phenotype, it's blended. And that's a special case. That's called incomplete dominance. That's when the in-between blends. So you're going to see scenarios like that this week where you don't have complete dominance, you have incomplete dominance. You're going to see scenarios this week where instead of incomplete dominance and blended phenotype, you have situations where they're both physically seen. So an example of that might be, let's say, chickens. There are chickens that are black. There are chickens that are white. I'm going to go ahead and change the little letter for white. And then we have chickens that actually can inherit one of each. And therefore we see black and white on a chicken. So that's a little different than what we've seen before. Again, there isn't straight dominance. Instead, it's dominance of both alleles at the same time. So that's called codominance, and that'll be a different scenario. Oops. You guys are going to see situations where there are multiple alleles that can produce different kinds of phenotypes. So you're going to see scenarios like with human blood type. We have type A blood that humans can have. We have type B blood that humans can have. We have AB blood that humans can have, and humans can have O. And those blood types arise from a number of different gene combinations. People can get A blood when one parent gives them an A gene and another parent gives them an A gene. But they can also get A blood when one parent gives them an A and another parent gives them the gene for a recessive blood type known as O. So there becomes a hierarchy of dominance where A dominates over O. In B, they can have two of the B genes to get B blood type, or they can get a B and a recessive O, and B dominates over O. That's like another rule for blood typing. In AB blood, someone inherits one of each. That's codominance, where they're both going to physically be seen on a red blood cell at the same time. So that would be like the black and white chicken. You're going to have people with O blood, and the only way they get that is if they get both of the O alleles. The codominance, by the way, means that A and B are equivalent to one another. They don't overshadow each other. So that's a different kind of combination. A lot of human genetics is complex like this. It's not straightforward and simple like some of the problems we've seen already. Straightforward and simple is usually reserved for the simpler kinds of organisms. There aren't that many human traits that are inherited in a simple way. That's why if you've seen any human examples, they're kind of boring. It's like dimples, earlobes, freckles, tongue roller, like really boring stuff. But the more complex human traits like hair, it's complicated. Okay, what else will you see? You'll see codominance, incomplete dominance, you'll see multiple alleles, you'll see sex-linked genetics, which is genes that are on the X chromosome in the 23rd pair of chromosomes for an individual. So, you know, in the 23rd position, you either have an X chromosome twice, which would make you a female, or you have an X chromosome and a smaller second chromosome called a Y, and that makes someone a male. But these are genes that are located on the X chromosome that we're going to be tracking when we do sex-linked traits. So females will end up with two of the traits, but males will only have one. It's not a trait that's located on their Y chromosome. So without getting into all of it, that's going to be a little different too. And those are a little bit more complicated for students to figure out. So I'm introducing you to some of those this week. And then for extra fun, I give you guys combinations of some of those in a dihybrid. You say, okay, what are the odds of having this sex-linked trait and this codominant trait inherited together? So then the Punnett squares get complicated because you'll start to see genotypes written out in crazy ways where an individual has something crazy like that as their genotype. And that's different. We've not seen anything like that. And then you'll have to FOIL that and be able to keep it all straight. It's complex. So next Monday, we will go over the complicated genetics during our morning chat with one another. Okay. So as far as this week is concerned, it's pretty similar to last week. The organizer has some of the same stuff on it. I didn't open it up this morning to have it ready to show you guys, but the organizer pretty much looks exactly the same as anything that you've been doing so far. The My Connect page should not be used for anything other than submission. If you haven't seen the new way of grading, giving feedback just yet, make sure that you take a look at that. We created that last week and pushed it out to students kind of midweek last week. I have outlined it for you on my main My Connect screen here. And for convenience purposes, I also just linked the week-by-week schedule or organizer. Some students were having trouble finding it from one week to the next, so it's right there. It's also linked in the calendar, but it's right there for you now. And then this beneath that link is how we explain how we're going to do credit and no credit feedback, and then we made a little video to explain it to everybody. And then I've recorded this Zoom session today for those people that are choosing to sleep in. Thank you to those of you who came early this morning. And then we will be posting all of those Zoom recordings right into the link that I've provided here. It's like a page where I'm just going to link all the Zoom recordings that we've done so far. So I'll put today's in there. And then on the schedule for this week, which is opening slowly here, just make sure you guys are looking at that final column of deliverables so you know exactly what I'm looking for you to submit. And just for clarity's purposes, some students are still getting confused about what pages am I supposed to do? There's so much stuff here. There's so many videos because I walk you through how to do each type of complicated genetics. I made videos, and that's in the second column. And then there's other videos made by Amoeba Sisters, Khan Academy, and Bozeman. If you don't need those other videos, don't watch them. It's just further clarification in a more animated way than what I can make, because my video is just like, this is what you do, like we just sat through right now. But I did provide you a video to explain everything about how to do all the complex problems. And then there is a little bit from CK12 as well on non-Mendelian genetics. So basically for your deliverable this week, we want you guys to do CK12, and you can either take notes or you can do the reading guide that's been provided to go with the CK12. And then any other notes that you take along the way, you're going to be doing certain workbook pages like page 102, for example. And then I didn't specifically outline workbook pages that are complex genetics, but if you thumb through the workbook from page 102 on, there's a lot. And I've referred to those pages as I'm explaining in my videos. So you'll see lots of them referenced, but there's all kinds of pages. And then I give you, do I give you answer keys? I made this so many weeks ago, you guys, I don't even remember. I think I gave you answer keys for some stuff, but it's not written as answer keys. So it might be like what I did when I was explaining. If you find that you want an answer key and I haven't given you anything, please ask, and I can always like snap a photo and upload it. It's just not a 2-second process. It takes me a few minutes to do it. So there's a ton of blood typing stuff in here, like way too much. So you can probably just ignore a lot of that. There is a blood type lab that's linked here if you want to just check it out and see how someone would determine like who to give blood to based on blood types. There's also some colorblind sex-linked practice problems that we linked you to. I don't believe those are from the textbook, There's a little self quiz, and there's a color blindness thing. So there's a bunch of different stuff. But the only thing you're really turning in to me are any notes, photos of workbook pages, and that's that. And then I included, a genetics quiz on quizzes. So I'm going to add that to this organizer because I forgot to type that last night. Complete the genetics, and it's going to be on non-Mendelian genetics quiz posted here. And for some reason, my old links have now expired. That's the annoying part of quizzes. It like leaves a link valid for 2 weeks, and then after 2 weeks, it expires. So I will re-upload the link to join quizzes and make sure you're getting to the right place, even if you've already joined. And that way, once you're in my class, you can see the non-Mendelian quiz that I posted. I do look at the scores on those just to kind of see like, how are you doing? But I can also see if you reattempt it 5 times, so, or more. So just kind of be mindful of that. Like if you just keep trying the quiz over and over again to see if you can get a perfect score, I can see that you're doing that. So anyway, it's not something that's going in for a grade. It's not really anything more significant than I'm going to put it in the grade book as a not graded item, just so that we have a record of this is how the student did, this is that they did attempt it, yada, yada, yada. So if later somebody comes back and says, well, how did Jack do in biology when you guys were on distance learning? Like, oh, well, he did this, 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 and this, and now I have a physical record of it. So if you bomb it, then that's just feedback for you. Like, you don't understand this. You should probably check in with Mrs. Craig. Okay, so I'm going to do my normal tutorial in the evenings again this week. That's not going to change as far as I can tell. So I wait till my kids are in bed at 8 o'clock, and from about 8 to 9, you guys can check in with me if you start doing this work and you have questions on specific problems or whatever. It's kind of just like it's office hours, but it's kind of just like tutorial. It's optional. You don't have to come. But if you do have questions, that's a great time to check in with me. And it doesn't start on Zoom. It starts on Google Chat. And then if there's like a specific question that it's just a lot easier for me to explain, this way, then I'll open up a Zoom classroom and we'll bounce into it together and I can just explain it really quickly. So that's kind of how that works. All right, if you guys have any last questions, feel free to stick around and ask. We got about maybe 10 minutes left if we need it. And then otherwise, have a good day. Have fun trying out genetics this week and let me know if you need anything.